In 1996, a drug called ritonavir changed everything for people living with HIV. Before it existed, the diagnosis was essentially a death sentence delivered on a timeline. After it existed, the same diagnosis became something you could manage, something you could live with. 75,000 people across the United States were taking up to 20 of these capsules every single day by 1998. 2 years of flawless production. 240 consecutive batches that passed every quality check without a single failure. And then one capsule did not dissolve properly. The analysts followed procedure, shut down the line, destroyed the entire batch, deep cleaned the production facility, standard response to a contamination concern. The next day, it happened again. The capsules that were supposed to be clear were coming out white and cloudy. Scientists brought samples to the laboratory and looked at them under a microscope. What they found inside was millions of tiny needle-shaped crystals. Nobody had ever seen them before. They made a fresh batch of ritonavir in the lab to use as a comparison. That also came out cloudy. They tried again. Same result. Every single attempt produced the same white paste. Within a week, every tablet coming out of both the laboratory and the factory was wrong. Abbott Laboratories had no choice but to halt all production of ritonavir immediately, but they could not simply switch it off. Tens of thousands of patients needed these capsules to stay alive. They found an alternative, a factory in Italy. Production began there and every pill passed the dissolution test. Relief. But the relief did not last long. The Chicago team flew to Italy to understand what the Italians were doing differently. They compared every variable: pressure, temperature, humidity, the exact weight of every ingredient. Everything matched perfectly with what Chicago was doing. No differences, no explanation. When the Chicago team flew home, Italy called. Within days of the visit, Italian capsules were also failing. In a matter of 5 to 6 weeks, every location producing ritonavir had been contaminated with the same crystals. The scientists did not know how to detect it, did not know how to test for it, did not know what had caused it, did not know how to prevent it, and did not know how to get rid of it. This was not contamination in the traditional sense. The thing spreading was not a bacterium or a virus. It was the medicine itself, transforming into something that looked identical but no longer worked. To understand what was actually happening inside those capsules, you need to go back about 170 years to a furious scientific argument between two chemists who could not stand each other. Justus von Liebig was one of the most respected analytical chemists in Europe. He had personally developed many of the techniques that the field relied on. He was also, by most accounts, extraordinarily difficult to deal with, arrogant, impatient, quick to dismiss anyone he considered beneath him. He received a paper written by a chemist named Friedrich Wöhler describing a newly discovered compound. The paper stated its exact chemical composition, 1 atom each of silver, carbon, nitrogen, and oxygen. Liebig was furious, because he had already discovered a compound with exactly that composition, and when he put a flame to his compound, it exploded violently. Wöhler's compound reportedly just sat there and did nothing interesting. These could not possibly be the same thing. Liebig published a paper attacking Wöhler's work directly, called him a hopeless analyst, said he should go back and check his results. Wöhler checked his results, found no mistakes, published a second paper standing by his findings. Liebig published another attack. This went on for 2 full years, each man becoming more convinced the other had lost his mind. Finally, they agreed to meet in Frankfurt. They would replicate each other's experiments in front of each other and let the results settle it. What they found stunned both of them. They were both right. 2 compounds, identical atoms, completely different behavior. One was inert and stable. The other was one of the most shock-sensitive explosive substances ever identified. The explanation was that atoms alone do not determine how a molecule behaves. The way those atoms are connected to each other, the arrangement of the bonds between them, matters just as much. Liebig's compound and Wöhler's compound had the same atoms arranged differently, giving them entirely different properties. These are called isomers, but isomers were not quite what was happening to ritonavir. When scientists tested the white crystalline paste using infrared spectroscopy, a technique that identifies molecular structure by measuring how specific bonds respond to light, they found something unexpected. The spectrum of the new crystals showed the same peaks as normal ritonavir, same atoms, same bonds, but with tiny deviations. Something had shifted. To understand that shift, chocolate is actually a perfect starting point. Chocolate gets its texture from cocoa butter, a fat whose molecules can stack together in multiple different arrangements. 6 of them, to be precise. Each arrangement produces different physical properties. The dull, soft chocolate that melts immediately in your fingers is mostly what is called Form 4, a crystal structure with a melting point of around 27 degrees Celsius. The shiny, firm chocolate that snaps cleanly is mostly Form 5, with a higher melting point of around 34 degrees Celsius. Same molecules, different stacking arrangement, different physical behavior. This is called polymorphism. Different structural forms of the same compound. And unlike isomers, polymorphs have the same atoms connected by the same bonds. But the way those molecules pack together in a solid changes how the whole material behaves. You can switch chocolate between its polymorphs with careful temperature control. Heat it to around 50 degrees to melt all the existing crystal structures. Cool it down to 34 to encourage Form 5 nucleation. Raise the temperature slightly to eliminate the unwanted forms, then cool quickly to around 12 degrees to lock in Form 5. Done correctly, you get back your shiny, snappy chocolate bar. The crystals inside those ritonavir capsules were a polymorph, a new structural form of ritonavir that nobody had ever seen before, now called Form 2. And Form 2 was dramatically more stable than the original Form 1. Stability sounds like a good thing, but in pharmaceutical terms, more stable means less soluble. Form 2 ritonavir simply would not dissolve properly in the body. Taking it was essentially the same as taking nothing at all. So why could they not just do what chocolate makers do? Heat it up, reset the crystal structure, get Form 1 back. They tried extensively, and it never worked. The reason comes down to energy. Think of each polymorph as sitting in a valley on an energy landscape. To move from one valley to another, a molecule needs enough energy to climb the hill between them. In chocolate, the hill between Form 4 and Form 5 is small enough that controlled heating can push molecules over it reliably. In ritonavir, Form 2 sits in a valley that is dramatically deeper than Form 1. Once a molecule falls into that Form 2 valley, the amount of energy required to lift it back out is enormous, effectively impossible to achieve without destroying the molecule entirely. Form 1 was gone, permanently, but this still left the original mystery unanswered. Why did Form 2 appear in the first place? Nothing in the production process had changed. The hill between the two forms should have been too high to cross spontaneously. For the answer, you need to understand what happened to church organ pipes across Northern Europe during particularly cold winters, hundreds of years ago. Tin, the metal those pipes were made from, also has two structural forms. Above 13 degrees Celsius, tin exists in its familiar silvery solid state. Below 13 degrees, and especially in extreme cold, it can transform into a different form, gray, crumbly, less dense. As the gray tin forms, it occupies more volume than the silver tin it replaced, causing the metal to expand and crack from the inside. A pipe would develop what looked like dark lesions on its surface. Those lesions grew. The pipe would split apart. Other pipes nearby would develop the same symptoms. People at the time had various explanations. Many involved the devil. The scientific explanation is nucleation. Once a small amount of gray tin forms, it creates a surface that other tin atoms can attach to. That surface lowers the energy required to make the same transformation. It reduces the height of the hill on the energy landscape. The transformation becomes dramatically easier. It accelerates, spreads. The term for this phenomenon is tin pest, because of how it behaves like an infection moving through the metal. Exactly the same mechanism destroyed ritonavir. Once a tiny amount of Form 2 crystal appeared, and scientists still do not know with certainty how the very first Form 2 crystal formed, whether from a chance contamination or a microscopic impurity on a production surface. It acted as a nucleation site. Its presence lowered the energy barrier between Form 1 and Form 2. Form 1 began converting rapidly. Form 2, crystals broke free, became airborne, settled on equipment surfaces, on clothing. When the Chicago team traveled to Italy, they almost certainly carried Form 2 seed crystals with them on their clothes and skin, seeding that facility the moment they arrived. Soon, no production facility anywhere could manufacture Form 1 ritonavir at all. Abbott held a press conference 5 months after the crisis began. Their scientists stood in front of reporters and called what had happened a chemical and scientific impossibility. Not because it violated any law of chemistry, it did not, but because nothing in their understanding had predicted it or suggested it could occur. The executive who spoke described it plainly. A company's size and the collective intelligence of its scientists, he said, have no relationship to this problem. This phenomenon is unpredictable. We are, in some sense, the victim of bad luck. Form 1 ritonavir was never recovered. All attempts to manufacture it failed. Abbott eventually abandoned it entirely and returned to an older liquid formulation of the drug. The liquid version had worse side effects and not every patient could tolerate it. But it worked. The ritonavir crisis triggered an enormous regulatory and scientific response. Pharmaceutical companies now screen their compounds extensively for polymorphs before bringing drugs to market, spending hundreds of thousands to millions of dollars on that screening. We now know that more than half of all chemical compounds are capable of existing in multiple polymorphic forms. Ritonavir itself is now known to have at least 5. The warning that Abbott's representative gave at that press conference has never been fully answered. Next time, he said, it may happen to another drug that does not have a safety net. That drug has not appeared yet. But the chemistry that made ritonavir disappear has not gone anywhere either.